Next, enzymes. What about enzymes? Enzymes are proteins. All proteins are macromolecules or polymers. Therefore, enzymes are also macromolecules or biological polymers. And what is the special function of the enzymes? Catalysis. And catalysis means what? Decrease in activation energy requirement and increasing the rate of the reaction. And this is after decreasing the activation energy, the rate of the reaction can be increased. This is known as the catalysis, and the specific function of enzymes is the catalysis of reactions. Therefore, enzymes are considered as biological catalysts. Okay, enzymes are considered as biological catalysts. And enzymes are produced in living cells. Enzymes are produced in living cells. What about general characteristics? General characteristics of enzymes. First, most of them are globular proteins. Globular proteins. Most of enzymes are globular proteins and they are biological catalysts. They are biological catalysts, which means they lower the activation energy of the reaction and increases the rate of the reaction. And most enzymes are heat liable, which means they heat unstable. Heat unstable means heat sensitive. What happened after increasing the temperature, which means when the thermal energy is supplied, the proteins are denaturated. Enzymes are produced by proteins, therefore enzymes are also heat unstable, and their presence does not alter the nature of the properties of end products. Does not alter the nature of the properties of end products. And substrate specific, highly specific to their substrates. Right? We are considering general characteristics of enzymes. And most enzymatic reactions are reversible. Reversible means glucose plus glucose make maltose plus water. Then maltose. Maltose can be hydrolyzed into glucose plus glucose by the enzyme maltase enzyme. Then again, when two glucose molecules are connected, what happens? Maltose can be produced. So the enzymatic reactions are usually reversible. Reactions are reversible. So rate of the enzymatic activity is affected by rate factors. What are the rate factors? pH, temperature, substrate concentrations. Those are the factors affecting the rate of enzymatic reactions. pH, temperature, substrate. Then enzymes are not being used during the reaction. Enzymes do not alter the end product qualities, properties of end products, and enzymes are also not being used. And they provide active sites. Right? As this is the enzyme, there is a specific region which attaches the substrate. Right? This is called as the active site. Actually, the reaction is taking place in this site. And this is called as active site. And some enzymes need non-proteinous components to catalyze the reaction, which are known as cofactors. Cofactors are actually helpers of enzymes. Cofactors are helpers of enzymes. Other than that, Inhibitors. Inhibitors now they disturb the reactions. Inhibitors now they disturb the reaction. Inhibitors now they disturb the reaction. Factors. First pH, second temperature, third substrate concentration, fourth inhibitors. Inhibitors are bad friends of enzymes. And cofactors are good friends of enzymes. Next, we mention enzymes catalyze the reaction. Catalyze means what? Decreasing the activation energy and increasing the rate of the reaction. If this is the rate and if this is the reaction profile, take this lactose plus water into glucose plus galactose. And then what are the product reactants? Lactose plus water. What are the products? Glucose and galactose. And these reactants have some amount of energy. And these reactants have some amount of energy. Therefore, we can represent reactants like this, which means glucose plus water. So lactose plus water. Reactants. To do the reaction, some energy barrier should be passed. Right? This energy barrier we can represent like this. This energy barrier means the reactants should require some amount of energy, some amount of activation energy to start the reaction. After, if some reactant can pass this gap, pass this energy barrier, these reactants can become the products. Right? If they are unable to pass this gap, again they come back to reactants. Then this is the energy of products. Then this energy gap represents the activation energy without enzyme. When the enzyme lactase is applied, the enzyme decrease the activation energy requirement, which means they decrease this gap. So this gap represents the activation energy with enzymes. So this gap means EA minus EA dash, which means the decreased portion of energy by enzymes. I think you can understand what I am saying. This one is the enthalpy change delta H. Right? So then what about the mechanism of enzymes? What about the mechanism of enzymes? First, lactose plus water. Those are the reactants. And this always enzyme is larger than the substrate. Enzyme has the active site. Then imagine this is the substrate. Right? This is the substrate, which means lactose plus water, like substrate. Then before doing the reaction, enzyme is there and substrate is there. Then what happened? They are combined with each other. Then what happened? Then the structure looks like this. And this is the active site. Substrates attached to the active site to do the reaction. Then this is called as enzyme substrate complex. Right? This is called as enzyme substrate complex. Then the reaction is happening. After doing the reaction, products are formed. Then what happened? Enzyme is released. It is available for catalyzing another reaction, and those are the products. These are the products, which means the glucose galactose like products. This is the mechanism. is the enzyme. This is available. Enzyme is not being used during the reaction, and enzymes do not alter the qualities of products, end products. So this is the specific region of enzyme which is called active site. There is a mechanism. It is called as induced fit mechanism. Induced fit mechanism describes the nature of the active site. Before talking about induced fit mechanism, before discussing the induced fit mechanism, we should understand there is an old type of mechanism. This is out of syllabus, but you should understand the key-lock mechanism too. Then you will have a better understanding about the induced fit mechanism. So in the case of key-lock mechanism, this is the lock. Enzyme is the lock. Enzyme is the lock. Sorry, enzyme is the key. Substrate is the lock. Which means then the active site of the enzyme is exactly equal to the shape of the active site is exactly equal to the shape of the substrate. Like the shape of the key is exactly equal to the shape of the lock. But in the case of induced fit mechanism, they are actually made up of small molecules. They are subunits. Therefore, this shape of the active site should not exactly equal to the shape of the substrate. There may be some small differences. And when the substrate reaches this active site, they do some interactions, and the active site changes their shape according to the shape of the substrate. In the case of induced fit mechanism, the shape of the active site and shape of the substrate is not exactly equal. After reaching the substrate into the active site shape of the active site is slightly changed and made a proper shape to bind this substrate, which means active site induces the fittest shape to the substrate. It is called as induced fit mechanism. The shape of the active site is complementary to the shape of the specific substrate of enzyme, and hence important in the substrate specificity of the enzyme. Here, in the case of induced fit, in the case of key-lock mechanism, they say the shape of the active site is exactly equal to the shape of the substrate. But in the case of induced fit mechanism, they say shape of the active site is not always fully complementary to the substrate. As enzymes are not rigid structures, the interactions between substrate and active site may slightly change the shape of the active site so that the substrate and the active site become complementary to each other. It is the induced fit mechanism. Then what about cofactors? As I previously mentioned, cofactors are helpers of enzymes. Cofactors are good friends. Good friends to enzymes. They are good friends to enzymes. They are non-proteinous components. Non-proteinous components. they, this is the active site. They bind to a site other than the active site. Cofactors bind to a site other than active site. They help to increase the rate of the reaction. Some are tightly bind and some are loosely bind. Tightly and permanently. Some are loosely and reversible. Anyway, cofactors help to catalyze the reaction, which means cofactors help to decrease the activation energy and increase the rate of the reaction. Next, factors affecting the rate of the enzymatic reactions. What are the factors? Four factors we should understand. First, temperature. Second, pH. Third, substrate concentration. Substrate concentration. Fourth one, inhibitors. Fourth one, inhibitors. As cofactors are discussed, first we will start from inhibitors. Inhibitors are bad friends of enzymes. Some they disturb the active site actually. They disturb the active site. Which means they prevent making enzyme substrate complex. Which means they disturb the reaction. There are two types of inhibitors. Some are irreversible. Example, toxins, poisons. Some are reversible. Drugs against microbes. Drugs against microbes. And there is another type of classification. Inhibitors are two types. First classification, irreversible inhibitors and reversible inhibitors, which means irreversible now they permanently bind to the enzyme and permanently decrease the rate of the reaction. Reversible now, if the inhibitor is removed from the enzyme enzyme can again act. So inhibitors are two types. First, competitive inhibitors and second, non-competitive. Competitive and non-competitive. Competitive inhibitors, the shape of competitive inhibitors looks like the shape of the substrate, which means as this is the substrate, as this is the active site of the enzyme, this is the inhibitor, which means the shape of the inhibitor is equal to the shape of the substrate, which means the inhibitor competes with the substrate. Right? These inhibitors bind to the active site instead of substrate. Such are called as competitive inhibitors. Most of the competitive inhibitors are reversible, which means when the inhibitor is removed, enzyme can get back to normal. Non-competitive now, this is the active site, this is the substrate, and this is the inhibitor. They bind to a site other than active site. When the inhibitor is bound what happened? The shape of the enzymatic active site is changed like this. But this is permanent. Even we cannot replace the inhibitor. Therefore, most non-competitive inhibitors are irreversible. Most competitive inhibitors are reversible. Most. Next, allosteric regulation of enzymes. Allosteric regulation of enzymes. I'm sorry. We have three factors. We have discussed three factors. We finished inhibitors. Next, temperature. Usually, this is the temperature. And this is the rate. Usually, the temperature of the system is increasing. Temperature of the system is increasing. Rate of the reaction is usually increased. While number of molecular collisions are increased. But after some, this is the optimal temperature. Optimum temperature. Which means this temperature shows the best action of the enzyme. And then after passing this temperature, enzymes are made by proteins. Proteins may be denatured by high temperature. After passing this temperature, proteins are denatured, which means enzymes are denatured. Then the rate is decreased like this. This is not an asymmetrical one. Most of the human enzymes have optimum temperature around 35 degrees to 40 degrees Celsius. This is the temperature effect. And what about the pH effect? What about the pH effect? This is the pH and this is rate. pH has symmetrical graphs. Sorry. Some enzymes of the body have optimum means the optimum pH means the pH of some enzyme which can perform the highest action of the enzyme, highest rate of the enzymatic reaction. This is the optimum pH of acidic enzyme like pepsin in the stomach. This is the optimum pH of alkaline enzyme, which means pancreatic enzymes. This is called as pH range. pH range means in this range, in any pH, between these two pH, any pH can affect the enzymatic reaction. pH range and optimum pH. Next one, substrate concentration. Substrate concentration. And rate of the enzyme. Imagine we have 100 molecules of enzyme in this beaker, and we add substrate molecules one by one, 1, 2, 3, like this. Then what happens after adding substrates? For every substrate molecule, enzymes are available. Therefore, the rate increase like this. But we pass in 100 substrate molecule, there are no enzymes available. All the 100 enzymes have bound with these substrate molecules. Then what happens? first enzyme molecule has to retain for another enzyme molecule, which means these enzymes do the make the substrate enzyme substrate complex and do the reaction and release products. Then again, the enzyme is available for another reaction. Therefore, the 101st substrate molecule have to retain. Therefore, after consuming all the substrate, all the enzyme molecules, the rate is a constant. Therefore, the shape looks like this. Mechanism, induced split mechanism we discussed. Cofactors. Hurry. First, temperature. Increasing temperature increases molecular motions. Therefore, speed of moving molecules of both enzymes and substrate will be accelerated, which means their number of molecular collisions will be increased. This will enhance the colliding probability. Colliding probability is increased, and more collision between the enzyme active sites and substrate molecules generate greater chances for the reaction to occur. This can continue up to a certain point, after which there is a rapid decline in enzymatic activity. This is the optimum temperature. Optimum temperature means the temperature which shows the maximum rate of the reaction. Most human enzymes, the optimum temperature is around the body temperature. But some bacteria, especially in hot springs, depending on their environment, optimum temperature may vary. When the temperature increases beyond the optimum temperature, what happens to the rate? Rate decreases. After passing the optimum temperature, there is a high temperature. We say the system has the high temperature. Therefore, hydrogen bonds, ionic bonds, other weak chemical bonds of the enzymes may be broken. Enzyme is denatured. This will result in change in the shape of the enzyme active site, will alter the complementary nature of the atom. Shape of the active site is changed. Therefore, complementary binding to enzyme active sites and substrate molecules will be prevented. This is called as denaturation of enzyme molecule. Therefore, the rate is decline when the temperature is increased beyond the optimum temperature. Stops complementary at certain temperature, although rate of collision will keep on increasing. pH graph is a symmetrical graph. Enzymes function most efficiently with a certain pH range despite maintaining temperature of the environment constant. There is a range, pH range. pH range means the narrow range of pH which a particular enzyme catalyzed reaction takes place. Optimum pH means the pH which shows the highest rate of the enzymatic reaction. Most enzymes, optimum pH range is 6 to 8, but there are some acidic pH as the optimum pH for pepsin like stomach enzymes and trypsin. Now, optimum pH is pH 8. Substrate concentration. Increase in substrate concentration increases the probability of collision between the enzyme and substrate molecules with correct orientation. However, the enzyme molecules will be saturated. As I previously mentioned, all the 100 number of molecules of enzymes are saturated with active sites. What happened? No any enzyme molecules are available to catalyze the new substrate. However, the enzyme molecules will be saturated after a particular concentration, and therefore there will not be any further increase in the rate of the reaction. Inhibitors we discussed, two classifications, irreversible and reversible. Again, competitive and non-competitive. The final topic of enzyme, allosteric regulation of enzymes. Allosteric regulation means, allosteric is a special name to explain the regulation of enzymes. The enzymes are, as we previously mentioned, inhibitors and cofactors can affect the function of the enzyme. As cofactors are good friends, as inhibitors are bad friends. Here, in the case of allosteric regulation, we are talking about some good regulators and bad regulators of enzymes. Here also, the regulation of enzyme is done by regulators. In many cases, the molecules that naturally regulate enzyme activity in a cell behave like reversible non-competitive inhibitor. Here, regulatory molecules we called as activators or inhibitors. Good friends na activators, bad friends na inhibitors. Regulatory molecules bind to specific regulatory sites elsewhere, which means other than the active site of the molecule via non-covalent interactions and affect the shape of the shape and function of the enzyme. It may result in either inhibition or stimulation of an enzymatic activity. Inhibition is done by inhibitors, stimulation is done by activators. First allosteric regulation type, allosteric activation and inhibition. Simply activators, when an activator is bind to the enzyme, the reaction is stimulated. When an inhibitor is bind to the regulatory site or active site is the site which the regulator binds. Okay, as this is the enzyme, as this is the active site, when the regulator is bind to this site, this site we called as the regulatory site or allosteric site. Regulatory site or allosteric site. Imagine this is the substrate. Black one is the substrate. Red one is the regulator, good or bad friend, which means activator or inhibitor. Then this is the active site. This is the regulatory or allosteric site. Allosteric site means what? The site which the regulator binds. When an activator binds with regulatory site, when an activator binds with the regulatory site, what happens? The enzyme is stimulated, which means stabilizes the shape with functional active sites. Whereas the inhibitor binds with the regulatory site, it stabilizes the inactive form of the enzyme. Then enzyme is, usually enzymes have the tertiary structure. Most enzymes have this tertiary structure. But in the case of quaternary structure, two or more polypeptides, two or more subunits are made to form the structure. Here we are talking about molecules which has more than two subunits. Subunits of enzyme arranged in a way through which they transmit the signals quickly to other subunits. Once one subunit is taking the signal, easily it can pass to the next subunit. Through the interaction of subunits, even a single activator or inhibitor molecule that bind to one regulatory site will affect the active site of all subunits. Example, imagine the cell needs more ATP. The cell needs more ATP to perform an active process. Imagine the cell needs more ATPs. Hari, the cell need more ATPs. Then what happened? One ADP molecule come close and it converts to the ATP. Hari, it connects with an inorganic phosphate and take energy from the cellular respiration and make ATP molecule. Once one ADP molecule is doing this process, which means once one ADP molecule is produced, one ADP molecule produces a ATP molecule, then other ADPs are come close. Hari, other ADPs are come close and more ATPs are formed. Therefore, ADP is, it starts the regulation, which means ADP can be acting as a activator. Hari. ADP function as allosteric activator, bind to the enzyme and stimulates the production of ATP by catabolism. Hari. The cell needs more ATP. Hari. No ATP available. One ADP molecule starts the reaction. Then other ATPs also convert to ATP. Then ADP initiates the reaction and it stimulates others also. Then ADP can be taken as an activator, allosteric activator. Allosteric activator. Once more enough amount of ATP is produced, no energy requirement further. Then ATP stops the reaction. Then ATP can be considered as an inhibitor. Can you understand what I'm saying? At the initial point, the cell needs more ATP. Then ADP starts the first ATP formation and other ADPs come close and other ADPs make more ATP. Then the energy requirement is fulfilled. After making more ATP, after fulfilling the energy requirement, no more ATP is required. Then ATP acts as an inhibitor and stops the reaction. Next, cooperativity. Cooperativity looks like this. You know, the hemoglobin molecule has 4 subunits. 4 subunits. Each subunit has a single heme group, and a single heme group has a single ion. Then 1 or 2 molecule can be bound to a subunit, which means overall 4 O2 molecules, 4 oxygen molecules can be bound to one hemoglobin molecule. Hemoglobin plus 4O2 reversible reaction and make oxyhemoglobin, HbO8. Imagine this is a free hemoglobin molecule. It is free of oxygen. No any oxygen is bound here. The first subunit tries making the first subunit tries combining of oxygen. It is so much hard work. Which means first attempt is so much hard. So first subunit, anyway the first subunit, after doing a hard work, first oxygen molecule is bound. Then other sites can easily bind their oxygen molecule. Bind in one substrate molecule and can stimulate binding or activity of other active site. Thereby increase the catalytic activity. Example is binding of one oxygen molecule to one binding site increases the affinity for O2 of the remaining binding site. Feedback mechanism inhibition means a metabolic pathway is stopped by the inhibitory binding of its end product of a process of an enzyme, which means in the previous case, initially no ATP, ATP requirement is produced. Then one ADP make ATPs, more ATPs. After fulfilling the ATP requirement, ATP can be acting. ATP can act as an inhibitor. ATP can stop the reaction. In feedback mechanism, a metabolic pathway is stopped by the inhibitory binding of its end products of a process to an enzyme, thereby limit the production of more end products than required and thus wasting chemical resources. Feedback inhibition is an essential process, regulates the end products and produced in metabolism. ADP function as allosteric activator and stimulates the production of ATP during the catabolism. In ATP supply exceeds the demand, catabolism slows down as ATP molecules function as allosteric inhibitor. Energy needed for all living processes is obtained directly from ATP, mainly produced by a process called cellular respiration in living cells. So that's all about theory of enzymes, and we will look past papers. Role of enzymes. Which of the above graphs best shows the effect of relative humidity? And this correct as temperature, not relative humidity. It should be correct as temperature. And so the third graph can be taken, asymmetrical graph. First one is suitable for pH. First one is a pH graph. Symmetrical graph is a pH graph. This is a temperature graph. Question 2 and 3 are based on the following graphs. 2003. Which of the graphs shows relationship of an enzyme and pH? Answer number C. 3. Relationship between rate of an enzyme reaction and substrate concentration. In then 2005. Which of the following statements is incorrect regarding enzymes? Enzymes are made up of amino acids. Correct. Enzymes are proteins. Proteins are made by amino acids. Correct. Enzyme reactions are reversible. Correct. Enzymes do not alter the nature of end products of the reaction. Those are general characteristics of enzymes. Some enzymes contain non-protein parts in their structure. Correct. Such are called as cofactors. Cofactors. Good friends. Enzyme molecules are usually smaller than the substrate molecule. No, enzyme is always larger than the substrate. Always enzyme is larger than the substrate. 2012, competitive inhibitors stop an enzyme activity by. Usually inhibitors are two types, competitive and non-competitive. And in the case of competitive, the shape of the inhibitor is complementary to the shape of the substrate. Therefore, inhibitor competes with the substrate. Okay. Competitive inhibitors bind to the active site instead of the substrate. Change in the shape of the enzyme, incorrect. Shape of the enzyme, incorrect. Combining with the substrate, no. Compete with the substrate. Blocking the active site of enzyme, yes. They bind to the active site. Combining with products of the reaction, no. Disturbing peptide bonds of the enzyme, no. Therefore, answer is number 2. 2020. Question number 7. Which of the following is a characteristic of enzymes? They do not alter the nature of end product. Yes, general characteristic. They increase the activation energy of reaction? No, decrease. Decrease activation energy. They are not substrate specific. No, they are substrate specific. A small amount of enzyme is used up during the reaction. No, none amount of enzyme is used up. Any part of enzyme molecule can catalyze a reaction? No, only active site can catalyze the reaction. Then 2022, question number 5. Activators affect the function of enzymes by binding to active site through covalent bonds. No, usually this is the enzyme and this is the active site. This is the regulatory site. Regulatory or allosteric site. A site other than the active site. And regulators are two types: good regulators, activators, and inhibitors. Activators affect the function of enzyme by binding the active site through covalent bonds. No, the site other than the active site. Shape of the active sites of enzyme changes due to temperatures higher than the optimal level. Yes. The process we called as the denaturation. The process we called as denaturation. Third one. Many competitive inhibitors bind the active sites of enzymes irreversibly and change their shape. No. Inhibitors are two types: competitive and non-competitive. Most competitive enzymes, most, not all, remember that, not all, most competitive inhibitors are reversible. Most non-competitive inhibitors are irreversible. Therefore, incorrect. Toxins bind to enzymes reversibly through covalent bonds. No. Toxin, poison are irreversible inhibitors. Coenzymes are proteinous components. No. Cofactors are good friends but non-proteinous components. Non-proteinous components. – 2023, question number 5. 2023, question number 5. In allosteric regulation of enzymes, regulatory molecules bind reversibly to the active site of the enzyme. No. Regulators bind to the regulatory site. Regulatory or allosteric site. Right? The site other than active site. As this is the active site, this is the allosteric site. Okay? Regulatory molecules bind to the enzyme via non-covalent interactions. Correct. Non-covalent interaction. An activator molecule that binds to a particular subunit will affect the active site of that subunit only. No. Okay? That subunit only. No. Next subunit can pass signals to each other. Fourth one, inhibitory molecules affect the function of enzyme but not the shape. Both shape and function. ATP functions as an allosteric activator. No. ATP is an allosteric inhibitor. Activator is a DP. ATP is an allosteric inhibitor. ADP is an allosteric activator. That's all about enzyme.